This is Ved Prakash. Welcome back to my channel. After a long time, I am making a video series on clinical research. So today we are going to discuss about clinical research. So what is clinical research? Clinical research is a field of healthcare science which deals with research on human beings. So it is not limited only to drug. It is also research on medical device or laboratory products or any diagnosis products. So this is we will discuss about clinical research today. So clinical research, the study is divided into two types, that is observational studies and experimental studies. So first is observational studies and second is experimental studies. So what is observational studies? So observational studies are like observing a cohort or a group of people. It can be retrospective or prospective. But what is experimental studies? Experimental studies are nothing but intervention where the drugs are actually given to the subject. So we can directly say clinical or clinical trials. So experimental studies are nothing else clinical trials. So after this we are going to discuss about what are the various drug development process in clinical trials. So drug development process. So for a drug which is discovered in a laboratory to come into market, it takes around 8 to 10 years and 100 to 500 billion US dollar is spent on a single molecule discovery. So what are the process of drug development we'll discuss today. So first, when a drug is discovered in a lab or synthesis, first synthesis of a molecule, then it will go to like QSAR, qualitative structural activity studies and all, and a lead compound will be discovered, a lead compound. So then this lead compound will go for preclinical studies. So we can divide this drug development process into different phases. So here we can say this is preclinical. Preclinical, that is humans are not involved in this phase, preclinical. Here the synthetic product or a lead compound which is our drug of interest will be go for preclinical studies, that is called animal studies. Animal studies. So in this animal studies we'll go for evaluation of toxicity, safety and efficacy. So toxicity and safety of our drug in animals. So that's why it is called as preclinical. So after preclinical, a pharmaceutical company or a sponsor will file a IND application, that is called as IND, that is investigational new drug, IND application to FDA, so that FDA will review it whether any toxicity is there or not, safety, what is the safety and effectiveness of a drug, and whether to proceed for clinical studies. So after IND application, that FDA approval, our drug will go for clinical studies. So here after preclinical we'll go for clinical part. Now this clinical part is divided into 4 parts or 4 phases, that is 4 phases of clinical trials, that is phase 1, phase 2, phase 3, then we'll go for phase 4, we'll discuss later. So what happened in this phase 1? After just submission of IND, all the reports of animal studies will be submitted and reviewed by FDA. Once it is approved, then we'll go for phase 1 study. In phase 1 studies, we'll go for like healthy volunteers or healthy subject. Here we say human who are participating in clinical trials are a subject or volunteers. So healthy volunteers, like around 20 to 80 healthy volunteers are taken, and here we go for safety and efficacy. So pharmacokinetics, PKPD, pharmacokinetics, pharmacodynamics studies are done, safety and toxicity studies are done in healthy volunteers, and here this phase will determine whether this drug will go to phase 2 or not. So here we go for pharmacokinetic, pharmacodynamic studies, safety and efficacy studies. So after phase 1, then if it is done, the drug is good, it is showing therapeutic effect, then we'll go for phase 2. Phase 2 is a like larger studies, like we can go for 100 to 500 patient. Here in phase 1 we are taking healthy volunteers, but here phase 2 we will take the patient. Suppose our drug of interest is diabetics, so here we'll take, from here onward we'll take the diabetic patient. So inclusion or exclusion criteria will be there, and they will be included in the trial. Okay, and here this phase is also known as therapeutic exploration. Therapeutic exploration means we explore the drug whether it is effective. Here also same effective, that is safety and efficacy is evaluated, and here that's why it is called we explore therapeutic exploration. And next, if everything is good, then our drug will go for phase 3. What is this phase 3? Phase 3 will take the large population, that is will take around 1000 to 3000 patient. Same, if our drug of interest is diabetics, then we'll take diabetics patient. Only in phase 1 we'll take healthy volunteer. Further, phase 2, phase 3 will take the patient according to the drug of interest. So here this is called therapeutic confirmation. This is also known as therapeutic confirmation, because we will confirm whether the drug is showing effect or not, whether we'll even consider the drug regimen. So here phase 2 will go for dosage, like what is the drug regimen. Suppose 100 mg is effective BID, that is twice a day, or QD every day. So drug regimen. So here exploring, here we are doing the experiment, but here therapeutic confirmation. That's why in phase 3 we take large number of people, because as we have sufficient document to satisfy that yeah, our drug has passed animal toxicity studies, phase 1, phase 2, we can go for therapeutic confirmation that our drug is having a effective therapeutic index, a safety and efficacy, as well as no toxicity. Okay, so this is called therapeutic confirmation. After phase 3, so I forgot to say one thing. Nowadays, like because of huge money is spent on clinical research, so there is another phase 0, that is called phase 0 before phase 1. Phase 0, that is called as microdosing. Here, as the cost of drugs and cost of clinical trials is more, so phase 2, here like less amount of dose is given to the healthy volunteers, and we can get the pharmacokinetics profile from that. So this phase 0 is not mandatory, we can directly go for phase 1, phase 2, phase 3, but for therapeutic exploration like pharmacokinetics and pharmacodynamics in microdosing, because of highly costly drugs, we can go for phase 0, that is called microdosing. So now, as we discuss about phase 1, phase 2, phase 3, our drug is having, we have confirmed that our drug is having effect in diabetics patient. So therapeutic confirmation, in confirm the drug is effective, safety and efficacy. So now after that we'll go for phase 4, the last phase of our drug development process in clinical research. So before going for phase 4, again same how we file an IND application to FDA. Again, we have to file an NDA application to FDA. So before going to this phase 4, this is, we can say, phase 4. Phase 4. Before going to phase 4, we have to file an NDA application. NDA application to same FDA or regulatory authorities of different countries. Like in India, it is CDSCO. Japan, they have their own regulatory bodies. In Europe, that is EMEA, European Medical Association. In US FDA, that is Food and Drug Administration. So this NDA application will be submitted to the regulatory bodies of respective countries. Then this NDA will review the entire file from even preclinical to phase 1, phase 2, phase 3. Then it will confirm or it will approve the drug, and our drug will hit to the market. So this phase 4 is nothing but the post marketing. This is post marketing surveillance. So here comes the preclinical drug starting from the laboratory and directly coming to the market. So from this process to this process, it will take around 8 to 10 years. So this is the long journey. A single drug from lab, it will travel to market, and lots of billions of money are spent for developing a single drug, approving by FDA. So this phase 4, what is there? Suppose this is called phase 4. Why? Because phase 4 is also called as pharmacovigilance or post marketing surveillance, because our drug is in market and it is having a wide range of effect. Here we are selecting only 1000, 2000 subject, but here it will go to market, like it will go and distribute it to entire globe, because here we don't know that a long term toxicity may occur long term. Or some drug, here we have selected suppose a European population, but that same drug may not be effect in Chinese population or Asian population. So here we see the adverse event, a long term adverse event, and we report to the FDA. If the adverse event or toxicity is high, then the FDA has full power to ban or withdraw the drug from the market. So here we discuss the entire process of drug development in clinical research. I hope this video is useful to you, and a further more videos series in clinical research is coming, and so please stay tuned with us and don't forget to like and subscribe my channel. Thank you.